As we have seen in our previous module that the process organisms can be collected from the culture banks. But there is a limitation. That is very easy to collect, and that can be having a very low cost, because either they donate that microorganism, either that culture, either they have a very little bit shipping cost. But if we want to exploit any microorganism for the industrial production according to the local environment, so it is the best that if we utilize the natural habitat to isolate our desired process organism. So in this module we will talk about that what will be our choice. The ideal isolation procedure commences with an environmental source. Frequently we mostly use the soil, which is highly probable to be the rich in the desired types, is so designed as to favor the growth of those microorganisms possessing the industrial importance or industrial important characters, that is industrially useful characteristics, that are used as a selective factor and incorporates a simple test to distinguish the most desirable type. So the selective pressure may be used in the isolation of the organism which will grow on a particular substrate in the presence of a certain compound or under the conditions, under cultural conditions adverse to the other types. So isolation criteria basically totally depend upon the techniques and the way how we are isolating and what is our main object. So in this slide you can see that there are the step by step what are the parameters we have to adopt for the isolation of any microorganism. So the first one, that we have to isolate the microorganisms from the natural source or natural habitats accordingly as our need. The next one, after the isolation, then is the identification of the desired microorganism. If we isolate and we know that have the potential to produce our desired product, then we know what is that organism. So then we characterize that microorganism. Then we screen, because if there are so many strains, then we select the hyper producer of that. And then if there is any possibility, we develop that strain for maximum production. And then we run the fermentation process. So these are the different steps that we have to go for the fermentation for better production. So as concern the different natural habitats and the sources, if we want to isolate some thermophilic microorganisms which we called as thermophiles, then our habitat should be the hot springs, thermophilic vent or the compost piles. As concern the alkalophiles, high salinity soils and the wells are the suitable habitats to isolate such kind of the microorganism. As concern the acidophiles, pine growing site, fruits, etc., these are the habitats. We want to isolate the osmophiles or halophilic microorganism, then the sea or different oceans or some high salinity lakes we can use. As concerned in Pakistan, we can have the suitable habitat of the Cura mines. As concerned the yeast, we can have the vineyard, etc. If we want to have a phosphate solubilizer microorganism, then agricultural soil, rhizosphere is the best choice. If we want to isolate the organism having the better potential of protease, then the dairy industry meat processing unit where the meat or protein substrate is in abundant. So if we want to isolate the microorganisms that have the potential to produce the cellulases, pectinases or xylanases, then is the choice is agriculture soil, paper and pulp industry, waste disposal areas of fruit use, plants, grain market. So according to our desire, we can select different habitats. So accordingly, we can isolate the organism having the desired characteristics. But as concerned that how we isolate, as concerned the isolation of the organism, the most applied method is the dilution method. So if we collect any sample either from the hot spring, either from the soil, either from the lake, that can be having a number of microbial load. So in order to get the isolated colonies, we first have a dilution of that. Suppose if we have a 1 gram of the sample, we diluted into the 100 ml of the sterilized water or a dilution buffer, and then we have the 9 ml in each tube, and then we add 1 ml and then mix it well, and then continuously transferring 1 ml to the further test tube. We have the dilution, and we pour on different plates. You can get different the microbial colonies on each plate. So as the dilution will be increased, then the number of colonies will be less. So at the end, if we go beyond 10 raised to power minus 5 or minus 6, then we have a 1 or 2 isolated colonies. Sometimes, as we have seen, that there is not a sufficient amount of organism in any sample, then we have to the enrichment cultures. We have to add the extra nutrient into the sample and then to multiply in the same environment, and then we try, and then we have the isolation as we called as a dilution factor. So as concerned the tenfold serial dilution on a natural or enriched sample and its inoculation on a selective medium is one of the most practically applicable procedure for initial isolation of the desired and all types of the microbes from the different samples, which give the mixed microbial population most of the time. So when we isolate, there is a chance of a less chance of the having a pure culture. So if we have isolated colonies, then we can have a pure culture. Otherwise, first of all, we have to adopt the isolation process, but followed by the pure culturing of the substrate, pure culturing of the microorganism.